Good morning. Good morning, everyone. Can you hear us? Can you write in the chat if everything is okay with the audio? I guess they have to hear us. Steph, you can hear me, right?
I can hear you.
Yes.
Ah, nice. Okay.
Perfect. Okay. So welcome to the Women in Neuroscience Symposium. We're very, very happy that so many of you decided to join us today. And we organized this series of talks as part of the initiative by the Spanish Society of Neuroscience in order to celebrate the International Day of Women and Girls in Science, which is today, February 11th. My name is Nadia Paraskevoudi, and I am a PhD student here at the Brain Lab at the University of Barcelona. And we, the PhD students of my research group, there are many more actually that do not appear right now on stage. These are the backstage heroes. We have organized this event for you. And we have compiled a great program of talks by female neuroscientists from around the world. And we will start with the talks by the researchers in the early stages of their careers with the PhD students, and then we will be gradually moving up the academic hierarchy, let's say. Our keynote lecture at the end of the symposium will be Dr. Sonia Kotz from Maastricht University. And after the scientific part of the day, we will have a roundtable discussion where the speakers will discuss their experiences of being women in a male-dominated field of research, which is cognitive neuroscience. Steph?
Yeah. Good morning. My name is Stephanie. I'm also a PhD student at the Brain Lab, and I just want to say very few things about the pragmatic aspects. So if you want to ask questions to our speakers, you can do this, but we're trying to separate the scientific part and the gender diversity discussion part a little bit. So first we're going to have the scientific talk, so people will speak about their work. And if you want to ask questions about that, please do after the talks. And later, if during the roundtable, you can also ask people questions. You can address specific speakers if you want about their experiences as women in neuroscience. Yeah, so we're trying to keep it separate. And if you want to ask a question, you can do that by clicking on Ask a Question at the bottom right of the screen. And then the platform publishes your question and other people can vote for the questions they like so that we can see which ones are the most popular questions. And then the chairs of the sessions can ask this question to the speakers. And if you want to speak about the event on Twitter, can you please use the hashtag WiN2021? Maybe we can go viral. And that's it, I think. Yes. Thank you very much for coming. And now Sonia is going to be the first session chair. Welcome, Sonia.
Can you hear me? Yes? No?
Yes, we all can hear you.
Okay, perfect. So good morning to everyone, and thank you all for being here today. My name is Sonia Arenillas. I'm a PhD student from the Brain Lab, our cognitive neuroscience research group located in Barcelona, Spain. And in this first part of the symposium, I'm going to introduce several PhD students that will present some projects that we are currently carrying out in the Brain Lab. In our laboratory, we work with electroencephalography to record the so-called AIEP, or auditory evoked potentials, which are neural responses that reflect the electrical activity of group of neurons to the presentation of auditory stimuli. To ensure that we don't run out of time during the talks, I ask you to please write your questions in the questions chat, and they will be addressed to our Brain Lab speakers when they have finished their round of presentations. So first of all, let me introduce you to Raquel Aparicio, who will present us the project of rock art landscapes, that is the emotion elicited by sounds in special places. Raquel, are you here?
She's just connecting.
Okay.
Hello, can you hear me?
Yeah, perfectly.
Okay.
So, Raquel, your time.
Okay, thank you. Let me share my presentation. Okay. So, greetings everyone. My name is Raquel Aparicio. I am a first-year PhD student, and I am currently working in the Art Soundscapes project, which is led by Professor Margarita Díaz-Andreu, and I am supervised by her and by Professor Carla Estera. So in this short talk, titled The Sound of Special Places, Neuropsychology and Emotion in Rock Art Landscapes, I am going to make a brief introduction on what is the Art Soundscapes project about, and I am also going to talk about the two research lines related to psychology and cognitive neuroscience that my colleague Samantha Lopez and I are working in. So the Art Soundscapes project, the aim of this project is to contribute to the understanding on how pre-modern societies were considering the relationship between acoustics and the sacred as components to produce rock art, which are human-made markings in natural surfaces like the one you are seeing in this picture. To this aim, the first step was, of course, to demonstrate whether there were differences in the acoustics between landscapes with rock art and similar landscapes but without rock art. So indeed, there is many evidence of that all over the world. So for example, some research has found enhanced audibility in shelters with rock art in San Serván, Spain, and others have found enhanced echoes in shelters with rock art in Baumbrun, France. Together with ongoing research on the project, these findings suggest that past hunter-gatherer societies were making rock art in special shelters or landscapes regarding the acoustics. Now, regarding the sacred, we know from ethnographical and anthropological sources that many communities make interpretations about the special sounds in rock art landscapes, leading them to believe that these places are connected to mythological beings. So, for example, we know that some societies, they go to these shelters, and when they talk, they think that the walls are talking back to them. Another example is this Lascaux cave in France. We know that this site is a shelter where many rituals and ceremonies took place, and recent fieldwork in acoustics has revealed that indeed this place is suited to this type of ceremonies because in some parts it resembles the acoustics of large concert halls. Together with similar findings all over the world, some have proposed the existence of universals in order to explain the importance of acoustics as a way of facilitating a communication between humans and mythological beings or ancestors. Now, to further examine the relationship between acoustics and the sacred, this project has a research line called psychoacoustics. Psychoacoustics is the study of the hearing system as a receiver of acoustical information, and importantly, this includes the participant's subjective perception of sound. In this research line, my colleague Samantha Lopez will determine which are the psychological effects when participants are presented with sounds with different acoustics. So although the target of the project is the acoustics of rock art landscapes, she has started to test some psychoacoustics methodology with a first online experiment where she's exploring whether participants experience music to be more expressive or beautiful when this music is played with the acoustical signature that is typical for that type of music. So specifically, in this experiment, participants are presented with two types of music, liturgical music and chamber music, and each one of these types of music can be presented in two conditions. First of all, they can be presented with the acoustics that are typical for that type of music in blue. So for example, the liturgical music can be presented with dry acoustics, which is what is common, but also its type of music can be presented with the acoustical signature that is typical for the other type of music, here in red. So for example, the liturgical music could also be presented with the acoustics of churches and cathedrals, which is typical of chamber music. For each one of these conditions, participants have to rate their emotions from 1 to 5 using a 9-item version of the Geneva Emotional Musical Scale, which contains items like joy, sadness, transcendence, or wonder. Although these results are preliminary, we are seeing some interesting trends, and this is that regardless of the musical type, participants are experiencing more transcendence when they are presented this music with the acoustics of churches and cathedrals, which are characterized by having more echoes or reverberation. The results of this first experiment will be important to know which items are modulated by the acoustics of sounds and not by the sound itself. And my colleague will use these items in order to directly assess the acoustics of rock art landscapes. To this aim, we have also built an immersive psychoacoustics laboratory, which you can see in this picture. It is a soundproof cabin with a three-dimensional sound speakers array, which provides with an immersive experience, which has been proven to enhance the emotional reaction of participants to auditory stimuli. Through a process known as auralization, this laboratory will allow us to play sounds as they would sound in real places. So this is with the acoustical signature of the places of interest. So in other words, this laboratory will allow us to bring the sacred soundscapes to the laboratory. So for this next experiment, my colleague will present to the participant naturalistic stimuli, so for example, thunderstorms, crickets, or wolves, in two conditions. First of all, these sounds will be presented with the acoustics of rock art shelters, and this will be compared with the emotional reactions elicited by these sounds with the acoustics of similar landscapes, but without rock art. My research line is called neuropsychology, and here we will test whether sound with acoustic properties of rock art landscapes induce brain activity patterns related to particular emotions, such as those elicited by altered or mystical states. To this aim, we will run a series of electroencephalographic experiments where we will modulate the acoustic properties of interest. So, to this aim, we started with the idea that sound, and especially music, are stimuli that can elicit exceptionally strong emotions, and they can regulate the mood of individuals. And even nowadays, for many rituals and ceremonies that we take part in, we use music to regulate the emotions and our moods. So, for example, for funerals, for military march, or for parties, we use different types of music, and its characteristic depends on the objective of each ceremony. In a similar manner, there is a commonality in the stimuli that is being used for eliciting a mental state that is similar to meditation or transcendence. And this stimuli is a very repetitive stimuli. So, for example, some meditation techniques use breathing or mantras, which is very repetitive. But also, many societies all over the world are using repetitive drumming in order to induce mental states similar to trance. So all these commonalities in the techniques and the stimuli being used have suggested that there might be some common biological basis. One biological basis that stands out among others in the literature is entrainment, especially for the auditory stimuli, auditory entrainment. This is a synchronization between the brain activity and the characteristics of sounds. So, for example, in this image, you can see a synchronization between the frequency of the oscillations recorded with electroencephalography and the frequency of a repetitive sound. This entrainment might have some psychological or behavioral consequences, eliciting these mental states similar to trance or transcendence or meditation. So in these next experiments, we will use auditory entrainment, and together with the enhanced emotional reactions to the sounds in rock art landscapes, we will be closer to a phenomenological understanding of the sacred soundscapes for pre-modern societies. And to end this presentation, I wanted to give a special thanks to all the Art Soundscapes members, especially to my supervisors, Margarita Díaz-Andreu and Carla Estera, and of course to my colleague, Samantha Lopez. Thank you for listening and for being here. And if you have any question, I will be more than happy to try to answer them.
So thank you, Raquel. It was really an amazing topic. And I remind you to write down your question on the chat, and they will answer at the end. They will be answered at the end of the 4 Brain Lab talks. So now I ask you to give your full attention to Stephanie Sturm, who is going to talk about prediction and memory processes in motor sensory interactions. So Stephanie,
Thank
welcome.
you. Thank you.
Your time.
I will share my screen. So, again, my name is Stephanie Sturm, and I'm part of a research line within the Brain Lab that is called Predictive Processes in Motor Sensory Interactions. We have 2 other PhD students, Marta Fontanaminos and Nadia Parasquevuri, and also research assistant, Tricia Cinca Tomás, and our principal investigator is Iria San Miguel. Perception is an active process. The same sensory information can be interpreted in different ways depending on the context or depending on the memories that you already have. And for example, sensory illusions illustrate the way that the brain is interpreting sensory information. It doesn't just receive it. And a very important factor for interpreting information is prediction. And predictions can be made on the basis of all sorts of things, but a very important source of prediction is motor behavior, because based on our movements, we can predict the sensory information that we think we're going to receive soon. And in fact, we have to predict that sensory information. And when we make, yes?
Sorry, we cannot see the presentation in full presentation mode. Do you have it in full
Yes.
screen? Maybe you can stop sharing and share again, because we cannot. You were moving the presentation?
Yes.
Oh, we didn't see anything moving.
I'm going to try again. I'm going to share entire screen. Allow. Okay. What about now?
Yes, now it's perfect.
Okay,
Thank
so
you.
this was the slide that was for sensory. The first slide here. It has this nice animation, and so yeah, motor information predicts sensory feedback, and so when we make predictions based on our movements and they're correct, then there tends to be a sort of cancellation effect where we perceive the sensory feedback that we receive in reality, we perceive it as a little bit attenuated, a bit less intensely. And when we make wrong predictions, and there will be a prediction error response, and that could be an element of surprise, or and this tends to direct our attention towards that stimulus that was unexpected. And a very key distinction that has to be made by the brain at all times when we process information is whether the source of the information that we're receiving is ourselves or an external agent. So one way to do this is to link your own movements with the other sensory input that you receive, and to understand how they are causally related. And so in our research group, we study sound processing. And so I'm going to give you an example from the domain of sound, but you could also say the same things about other sensory modalities. So, for example, when you play a piano or when you listen to music passively, you have different situations. In the case that you're playing, you're moving, And the movement is controlling the sounds that you hear, and the sounds are quite predictable because you have full control over the situation. You can just determine the timing and also the nature of the sounds that you're going to hear. However, when you're passively listening to music or to another sound, it doesn't have to be music, then you don't move, or if you move, it's not related to the sounds, and you don't have control over the sounds. They might still be predictable because you might be familiar with the kind of sounds that you're listening to, but you might not be it. So this can vary. So how do these different situations manifest? How can we measure how these things are differently processed by the brain? Self-generation effects is what we call the stuff that we can observe when we ourselves are the agents of stimulus. So on the behavioral level, we can, or on the perceptual level, we can observe attenuation. At least very often we can observe attenuation, where the self-generated sounds are perceived less intensely, less loudly as externally generated sounds. The relationship is a bit more complex than this, but I cannot go into a lot of detail right now, but other talks later on will, I think. And on the neural level, we have an analogous attenuation of ERP components to self-generated sounds. And we can also, for example, measure differences in pupil dilation depending on whether there's movement during a stimulus or not. And these are like simple observations, and we try to understand what are the mechanisms that drive these observations. And there are at least two very important factors that we have to take into consideration. One is based on predictions, and the other one is based purely on the presence of movement. So we speak about predictive forward models that cancel the effects of incoming stimuli, but we also have observed that mere coincidence between a movement and a sound leads to neuromodulation, and we call this like the halo of neuromodulation that surrounds motor acts, because even when there's no causal relationship between the movement and the sound, the brain processes differently the sounds that it hears while it presses a button, for example. And these are completely, well, these two factors, they come together and they influence the way we process or we distinguish between self and externally generated sounds. Then another question that we're asking ourselves in our PhD work is how these effects during sensory processing affect memory. And there's a phenomenon that is quite well known that is called the production effect, which is that spoken words are remembered better than silently read words, or melodies that you play on the piano yourself. You can remember them better than when you're passively listening to a melody. And also it has been shown that task-irrelevant button presses, so movements that are not causally related to the sounds, still affect memory of items and improve memory of items. And so we are trying to understand how these observations relate to the self-generation effects that we can see during sensory processing. So in summary, we have these different factors: unspecific neuromodulation due to the presence of movement, and motor-based specific predictions based on your movements, and also predictions in general without movement, and also the feeling of agency and the feeling of being in control of the stimulus that you're listening to, that together seem to make up both of these effects, or at least contribute to these effects, self-generation effects and the production effect. And we're trying to understand what the individual contributions are and how they work together. So we're trying to understand in our PhD work what the psychological and the neurophysiological mechanisms are behind these things and how they affect memory. And we try to study the relative contribution of movement sound coincidence, of predictability, and of control, the feeling of agency, to the phenomenon. So we have 3 PhD projects at the moment. The first one, Nadia and Teresa are working on, which is coincidence versus contingency. So they're trying to understand what is the effect of predictability or the lack of predictability versus coincidental co-occurrence of movement and sound, and how do these things affect processing and recognition memory. And Marta's working on memory structure, so she's trying to understand how the source of a stimulus, self or externally generated, affects the way that this sound is remembered and structured into how different sounds, different events are structured in memory. And the boundaries between different transition events. And my project is on agency and control. So I'm trying to understand the effects of I'm making decisions actively and feeling like the agent of a situation on decision on sensory processing, learning, and memory. The experimental paradigms that we use have normally an encoding phase where people press buttons and listen to sounds, or in my case, they move their eyes and they generate sounds with their eyes. And we manipulate the degree of contingency between the movements and the sounds, and also the amount of control that people have. And then we have a retrieval phase where people try to remember items, and this can be either recognition memory or order memory or associative learning. And we study behavioral measures like memory performance, and we also use ERPs and EEG, and we also measure pupil dilation to get a bit of as a measure of subcortical contributions. So in conclusion, we are trying to understand the link between self-generation effects and the production effect, or in other words, the effects on memory of the self-generation and sensory processing differences. Thank you so much for your attention. Thanks for being here. Please ask questions later, and you can also get in touch. These are our Twitter handles and the hashtag again. Thank you.
Thank you, Steph. So now we are going to move to Janina Puru. Stephanie, Natalia is going to make you out of the screen.
Bye-bye.
Okay, so Janina is going to talk about the role of genes in modulating brain potentials and how this is associated with speech and musical processing. Janina?
Hi, and welcome for everyone. I will, no, I'm sorry. Oh.
Can you share your screen?
Yes.
Yeah, perfect.
Okay.
Perfect.
Okay, hi, and welcome for everyone. My name is Janina Puru. I'm a PhD student in the Brain Lab Research Group, and I'm so glad to present you a brief resume about my PhD research, which is the role of genes in modulating brain potential associated with speech and musical processing. It is a fact that the individuals differ from each other in their ability to so process, which can be seen reflected in various contexts, for example, skill related to speech or musical ability. But it is unclear what role the genetic factor plays in explaining these differences observed in so processing. So, in order to answer this question, we must first understand how the auditory system works, how they encode the auditory signal into different levels of the auditory hierarchy, and then evaluate if the genetic variant can explain these differences observed in so processing. So how the auditory system works. At the anatomical level, the acoustic features are encoded and processed at a different level of the auditory hierarchy from the ear through the subcortical structure to auditory cortex. At the anatomical level, the auditory stimulus is converted from an acoustic waveform into an electrical signal, and this is encoded along the auditory pathway, and the overall signal registered on the scalp correspond of the sum of the all activity in the different subcortical and cortical stage. So, depending on its origin and its latency, the neural activity that we observe after an auditory stimulus occurs can be separated in three categories. The first one is the auditory brainstem response. This response is generated in subcortical structures, and we can see this response in the first 10 milliseconds after an occur auditory stimulus. The middle latency response, that is the response generated in the medial and geniculate body in the thalamus and the primary auditory cortex. And finally, the long latency response, that is the response generated in the cortical areas. It has been observed that not only the cortical structure contribute to the sound processing, but also the subcortical structure are involved or play a role in their processing. Oh, sorry. So the frequency following response is a kind of auditory brainstem response originated at the subcortical structure that imitates the incoming stimulus. This means that is synchronized with the spectrotemporal characteristic to the of an incoming sound. In this figure, we can see an example. This is an audio stimulus and the corresponding FFR, and we can see the EEG is synchronized to the audio stimulus, not only in the time domain, but also in the temporal spectral domain. The FFR has been very used to study the encoding sound because it's very sensitive to the context and the auditory's experience. For example, Musakia and colleagues found that the musician showed greater amplitude in their FFR in comparison with the non-musicians. In the similar way, Chrisman and colleagues found that the bilingual have more amplitude in their FFR response in comparison with monolinguals. So far, the literature suggests that the environment variables can influence the FFR response, but what happened with the genetic factors? Several studies in twins with behavioral measures have reported that the ability to process sound is highly inherited. For example, it was observed in different twin studies that the heritability between 50 to 70% in different auditory skills. However, at the neural level, there is only one study that relates some polymorphism of a gene associated with the serotonin uptake with the sound processing at the subcortical stage. This gene is the SLC6A4, and it's encoded for a transporter serotonin genes. These genes have two allelic variants in the promoter region, the short and long allele variant. The short allele variant is associated with reduced expression of the transporter genes. This resulting in a high level of the the serotonin available in the synaptic cleft here. And the long allele is related with increased expression of the gene, which cause more serotonin transporter and therefore less serotonin available here in the synaptic cleft. So the researcher create two groups based on their serotonin expression and compare how the compare how the subcortical sound encoding differ between them. So the result show that the low expression group in green have greater response in comparison with the medium and high expression group. That means that the sound encoding is enhanced on the subcortical structure when there is more serotonin available in the synaptic cleft. So, So this experiment lead us to ask ourselves if serotonin would also modulate more complex stimulus, and if maybe other genes could be also influencing so encoding. In this line, the purpose of my PhD project is to evaluate the role of genetic factors in the individual differences observed in processing of acoustic stimuli of different nature and complexity at a different level of the auditory hierarchy. Specifically, we want to identify genetic variant associated to the frequency following response and cortical response to different stimuli of different complexity. In addition, we want to evaluate the role of the attentional process on the genetic modulator of sound encoding. Also, we want to establish the relationship between the observed genetic differences with functional assessment. And finally, we want to establish the relationship between the different behavioral measures with electrophysiological measures. In order to achieving this proposed goal, we will carry out 3 EEG experiments with stimuli of increasing complexity. In the first experiment, we are going to use a simple amplitude modulated tone in a Robin standard paradigm. In the second experiment, the stimuli used will be a high-level OA designed in our lab, and to able to fully test the encoding of sound at the subcortical level. And finally, the third experiment is the brain encoding of running speech. This is the in this experiment, the stimuli are sample of a continuous speech from a male and a female speakers, and these samples are obtained from publicly available audiobook in Spain. In addition to the 3 EEG experiment, the genetic sample will be obtained through the saliva sample, and there are some candidate genes that we propose to the analysis. It's only a resume. And finally, we want to complement it with several behavioral measures that include tests about musical ability, bilingualism, some reading skill, spacing noise perception, and amplitude modulated discrimination tones. This is the plan of my PhD research. Right now, I'm collecting the data. I want to thank to my PhD supervisor, Dr. Mark Villa, and of course the research group, Brain Lab research group. Thank you, all of you.
Thank you, Janina. It was a very exciting topic and also a very nice presentation. Congratulations. So finally, our last Brain Lab speaker is going to be Marta Puertollano, who is gonna present us how is the encoding of speech sounds in newborns. So, Marta, are you here? I feel that you need to stop sharing.
Here yet, 1 second.
Yeah. Yeah, perfect. Yeah.
She should be here in a moment.
Sorry.
Hello, Marta.
Hello.
Good morning. Can you share the screen?
Yeah, wait a second. Okay. I can't find the...
In the top of your face, there's a button where there's a screen with an arrow.
No, I know that part, but then it doesn't allow me to share the presentation, the PowerPoint.
Then you can share your whole screen.
Wait a second. Mm. It says compartir tu pantalla en ventana de la aplicación. So there is where I need to find the presentation, no?
You need to share your whole screen.
Ah, okay, sorry. Okay.
Now, Marta, perfect.
Yeah, okay. Perfect.
Now you have to put it full screen, Marta.
Exactly. So,
All right, thank you.
it's yours.
Okay, hello everyone. My name is Marta, and I'm a PhD student at the Brain Lab Group, and my supervisor is Carla Cestera. And during this presentation, I will try to give you an overview on the encoding of speech sounds, which is a variable since birth, and the repercussions this may have in clinical settings.
Marta, sorry. Can you try to unshare and share your screen again so we can put the screen big?
Ah, so it's not in big. Okay. Is it now?
No,
No?
no, it's because it's your face and in the other side is the screen, but it's very small.
Ah, seriously? So it's half and half? Is it? Vale, okay. Because maybe it's not the screen what I need to... Sorry, eh? I really don't know what to do.
Maybe you try stop sharing your video and maybe we can see the presentation bigger. Oops, she went out. She went out of the... 1 second. I think maybe we can go with one question from one of the speakers, and then we can, while we are waiting. Raquel, are you here? Because there are a question for you, maybe is the most vote so far. With Marta, because it should be Natalia who invites you. Natalia is going out on the internet. So yeah, I cannot share anything. Nadia also cannot.
Hi again.
Now.
I'm gonna try it again, okay? Sorry.
Okay.
Now I can share it. Yeah.
Yeah, okay.
All right. Do you see it?
Wait, I will stop my video. Are you sharing now?
Yeah, can you see it in the full screen? No.
No. Even not in a small screen.
Okay. Okay, great.
Now it's perfect, Marta.
Can you see it now?
Yeah, perfect. Go on.
Okay. All right. So, first of all, once the baby is born, they come to the world with an auditory system ready to face the auditory environment. And we can take advantage of this fact evaluating the integrity of the auditory system at birth. And we're also using the universal newborn hearing screening, which is based in auditory brainstem responses and autoacoustic emissions. Basically, this screening has been implemented all over the world, and it has saved thousands of babies from deafness. The stimuli that it uses is a simple stimuli, and this is the response that we get from the brain. However, even a large number of babies have been saved from deafness. There's still a large number of babies that will successfully pass this screening and face neurodevelopmental delays related to their impairments in language and reading abilities. So if we change this simple stimuli for complex stimuli such as speech or music, we can get a different brainstem response, which is the frequency following response, and it's this one. And this response can give us information about the auditory neural processing of complex sounds, and it reflects the encoding of temporal and spectral sound features in the auditory system. Thanks to the literature, we know that it appears disrupted in neurodevelopmental speech and language disorders, and also in autism disorders, spectrum, sorry. Where deficits in timing and frequency encoding are also reported. But also, it can be modulated by musical training. It is sensitive to musical and language experience. So, considering the potential predictor power that the FFR may have and the possibility to modulate it by training, it's what motivated our group to study the FFR during the first days of life when the plasticity is optimal. We basically, as in line with Nina Kraus and colleagues, we basically imagine an ideal scenario where after getting the newborn hearing screening, the baby also gets an FFR, the newborn. And this would only take a few more minutes, and and it could be done at the hospital, and it could give us valuable information about their development so that they can be referred as soon as possible to developmental specialists. In line with this, our general aim is to disentangle if the FFR could serve as a biomarker of auditory encoding disabilities in speech sounds processing. And for that, our first objective was to understand if we can establish a normative database so that we can see the standard variability of the FFR response in normal population. And for that, my coworker Teresa, she recorded this DFFR to DA and GA stimuli in term newborns born healthy, and she did it at the hospital. And basically what she found was that most of the parameters showed a normal distribution and that similar responses were recorded to both stimuli, DA and GA. So once the normative database was established, our next objective was to understand if the FFR could serve as an indicator of literacy disabilities in clinical population. For that, 2 different studies were carried out, and the first one was focused on the lower 10% of the normal weight distribution. Those are the ones that are born small for the gestational age, and they were compared to the adequate to the babies born adequate for the gestational age. Their FFRs were recorded and using that as a stimulus. And basically what we analyzed were the parameters indexing the accuracy of pitch encoding. We focused on pitch because this is an acoustic cue that is essential for language acquisition in infancy. And the results showed a deficit in neural pitch tracking in the vowel region for the small neonates. This could be explained by the white matter affections that are already shown in term growth restricted neonates in the literature. The second study was focused on the upper 10% of the normal weight distribution this time, and those are the ones that are born large for the gestational age, and they were again compared to the newborns born adequate for the gestational age, using again the DA as a stimulus and recording the FFRs. And basically, the ones that are born large for the gestational age have been associated to metabolic and cardiovascular diseases, but evidence regarding neurodevelopmental outcomes is limited and inconsistent. So we aimed to bring evidence with this study in the existence of perinatal central nervous system dysfunction in this clinical population. And the results showed a deficit in pitch encoding, not only in the vowel region of the stimulus, of the response, but also on the consonant transition of the response. And this means that a disrupted FFR could give us valuable information to decide derivation of affected clinical cases. But before that, we also need more studies to be carried out. So the next step was to design and validate a new stimulus that could give us information not only about the pitch, but also about other important speech cues. For that, my coworker Sonia, she created a new stimulus, which is the OA stimulus, and is divided by two vowels, the O and the A, but also divided by a steady part of the fundamental frequency and a rising part of the fundamental frequency. And the aim was to characterize the voice pitch and also the format structure encoding accuracy. To validate this stimulus, she compared a sample of adults versus a sample of newborns. And basically the results showed a pitch tracking that is adult-like at birth, but the format structure encoding accuracy would be maturing in a frequency-dependent manner, suggesting that the encoding precision at higher frequency ranges is still immature at birth. This opens up the possibility to study these sound features that could provide a different information. So based on the significant differences observed comparing clinical versus normal population and using the OA, which we've seen, the OA stimulus that we've seen, we can study characteristics not mature yet at birth. Our next step was to ensure the predictive power of the FFR. And for that, a new project is being carried out now at the hospital, and basically is a follow-up where the FFR is recorded at 3 points of times at 0, 6, and 12 months of age. But also we add other techniques so that we can evaluate the maturational changes at cortical and subcortical levels in the auditory pathway. So for that, we add the neurosonography before the birth at 32 gestational weeks, and magnetic resonance imaging to evaluate their brain also, but after birth, at 0 and 6 months. But we also add some neurological scale so that we can also evaluate their neurodevelopment at 6 and 12 months of age. With all this, we would like to characterize the underlying neuropathology of abnormal FFR, and also to understand which parameters of the response, the FFR response, could have a stronger predictive power. With all this, even though more studies need to be done and more follow-ups need also to be done, I would like to emphasize on the potential role of the FFR as a possible clinical tool that is non-invasive, is cheap, and is easy to use, and it could be implemented at the hospital without interrupting the newborn's sleep. And it could give us the possibility to implement early preventive programs as early as at birth. And that's all. Thank you for your attention.
Thank you, Marta, for your presentation. It was really nice. So now we have 5 minutes for a question. Oh, Marta, I'm hearing me from your. Can you turn off your microphone?
Don't worry, I closed my window. No?
Okay. Bye.
Okay, bye.
So perfect. Now we have like 2 minutes for question because we were a little bit of delay with the technical problems. So I will ask one question to Raquel Aparicio, since she's the most vote, and the rest of 2 questions, they will be answered on the chat because, yeah, the next speaker needs to be here at 10. So Raquel, the audience is asking if you can give some brief explanation on how do you manage to apply the acoustic conditions of the archaeological sites to sounds.
Okay, thank you for the question. So the thing is that we go to the sites of interest. So for example, the project has gone to many places like Siberia or Catalonia, and there we play a sound, for example, a sine sweep, which is a sound that contains many audible frequencies. And this gives us the impulse response, which is something like the acoustical imprinting of the place. And this allows us to compile many acoustical parameters of interest. We run them with whatever sound that we want and put it in loudspeakers, for example. And in this way, we can make any sound like it would in the site of interest.
Okay, it's really interesting. So I see you now. Goodbye. And now moving out of the Brain Lab, I'm pleased to welcome Emily Thomas. She's a PhD student from the Birkbeck University of London in the United Kingdom. And Emily obtained her bachelor in psychology at the University of Lincoln in 2015, and then she studied her master in brain and cognition at the Erasmus University of Rotterdam. Hello, Emily. I continue presenting you. She's currently doing her PhD, focusing on how our expectation can guide and influence our perception about the world during the action. Today, she's going to talk about the neural sharpening of predicted action outcomes in the visual cortex. Before she starts, I just want to remind you to write your questions on the chat for the end of her talk. So welcome, Emily. Thank you so much for accepting our invitation to participate in this symposium. So it's your time when you want.
I'm just going to share the screen.
Yeah. I will stop my video.
Does that work?
Yes, working perfectly. So.
Okay, great. Okay, well, thanks very much for inviting me, and also just for the Brain Lab for organising this meeting. It's really nice. So I'm going to be talking about some work that I've done in my PhD on how action predictions influence sensory processing. So theories of prediction outside of action context suggest that we combine our expectations with the sensory input in order to to make perception more veridical. And this is often when we have ambiguous sensory input or in the face of sensory noise. These ideas are commonly termed in terms of Bayesian theories, in which we combine the prior with the likelihood in order to estimate the posterior. And therefore, this would result in upweighting of perception such that we perceive what we expect. So, for example, if we're in the context of like a snowy Arctic scene where there's a snowstorm, it would be more adaptive for us to perceive that it's a polar bear running towards us when the input is ambiguous because of the snowstorm, than, for example, an elephant, which is an unexpected animal in that context. So this theory would also propose that we sharpen the neural representations of expected events. And so, yeah, in general, perception is upweighted. And another example could be in the face of sensory noise, such as in this kind of Muni image here. Those of you who haven't seen this picture before will probably struggle to work out like what's there. However, if I give you some prior knowledge that this image actually contains a cat, when I show you the ambiguous image again, you're probably a lot more easily able to identify a cat in this image because you've combined your prior knowledge with your input to perceive the cat amongst the noise. So this is a bit contrasting to theories of prediction in action. As we've heard a bit already from Stephanie, cancellation theories would propose that expected action outcomes are suppressed. And then this process is thought to be adaptive because it's to make an unexpected events more informative. And so by making them more informative, that means that we can update our models of the world or correct our actions when an unexpected thing happens. So this would suggest that neural activity would be suppressed for expected events relative to unexpected events. So a more concrete example in action would be, so for example, if we want to go and pick up a cup of tea, we can predict properties such as its weight based on how full the cup is and how thick the rim of the cup is. And we can use these predictions to adopt the appropriate action in order to pick it up. So when we pick it up, if it is actually lighter or heavier than we expect, then we need to be able to prioritize these unexpected events in order to correct our actions so that we don't drop or spill the cup. And then these theories are thought to explain why we can't tick ourselves as well. So as I've kind of outlined, the two theories predict opposite influences on perception. And so the main question of my PhD has been to ask whether action predictions do influence perception in a distinct way to other types of prediction outside of action contexts. So one of the ways, so a lot of neural findings have shown support for cancellation accounts. So they then demonstrate this kind of expectation of suppression effect. So that is basically when self-generated action outcomes elicit a suppression of activity in brain areas relative to externally generated events. So, for example, in a study by Konstantina Kilteni, who we'll hear from later, she found that self-generated tactile outcomes elicit a reduced bold response in secondary somatosensory areas and cerebellum relative to external events, which are less predictable. And this finding has been found by a lot of other researchers, for example, Blakemore and Shergill and colleagues, all finding that self-produced tactile actions outcomes are suppressed relative to externally produced tactile outcomes. And this finding has also been shown in visual cortex as well. So for self-generated visual outcomes, this elicits a reduced response in primary visual cortex relative to externally generated outcomes. So these studies all provide support for the idea that expected events kind of reduce or suppress neural activity. However, there has been some studies outside of action context which have suggested that such a kind of expectation suppression effect or lower bold signal does not necessarily reflect a cancellation type mechanism. So, for example, this study by Peter Koch and colleagues, they presented two different pitches, so a high or low pitch tone, and this could predict with 75% validity the orientation of the grating, so either clockwise or counterclockwise. And then they want to look into primary visual cortex to see how the activity changes depending on whether it's an expected event or an unexpected event. So initially they find the same kind of expectation suppression effect where expected orientations elicit a lower bold signal than unexpected ones, and that's in 2 different tasks. However, they also performed a pattern classification which discriminated between the orientations of the stimuli, so they could discriminate between clockwise or counterclockwise stimuli. And they found that classification accuracy was superior for expected than unexpected events. It suggests that there's actually more signal relating to the stimulus orientation when it's expected than when it's unexpected. So in order to kind of work out what might be going on here, we can think about what kind of mechanisms might be underlying this data. So if you consider that there's multiple different units in primary visual cortex that are tuned to different edges of orientation, if you present a vertical edge like this, or like a grating, the activity pattern that you might expect to observe when you present this vertical edge is in blue here. So here you can see on both images that there's a larger neural activity for the units tuned to the vertical edges. And this is then kind of reduced as the units then prefer different orientations further away from the vertical edge. So under a cancellation account, if you presented this vertical edge and it was also expected, the pattern you would expect is in red here. So there would be a suppression of expected response. So all of the units kind of suppress, especially the ones tuned to the vertical edge, are suppressed relative to the baseline. This kind of reflects the typical expectation suppression effect. However, the Peter Koch and colleagues outlined an alternative mechanism which might underlie the data instead, and this is through a sharpening account. And so it could be that when you present this vertical edge and it's also expected, you observe this kind of sharpened neural response because you've pre-activated the vertical edge when it's expected. However, all of the units that prefer other orientations, the activity is then suppressed. So here you can kind of see how you would observe a general expectation suppression effect because the majority of activation is suppressed. However, only in the units really tuned to that vertical edge are sharpened here. And so this might be able to explain their data in terms of showing an overall suppression, but there was actually more information about the orientation information when they did the multivariate pattern analysis. So we wanted to work out or find out whether this kind of sharpening model could be applied to action predictions. So we adopted quite a similar paradigm where the outcome is a oriented grating. However, this time it was action that could predict the grating. So we presented participants with a training session so that, because this is obviously an arbitrary mapping, participants could learn that their index finger movement or little finger movement, so they would either do that or that, could predict the orientation of the gratings. This was 100% at training, and the task was just to answer whether it was clockwise or counterclockwise. And then in a test session, which was in scanning, so this training was the day before. So in scanning, we then degraded these contingencies so that we could manipulate expected and unexpected events. And so a third of the time, participants made an action and they were presented with the grating that they were expecting, the orientation that they were expecting. A third of the other time, they were presented with an unexpected orientation, and the final third, they weren't presented with any stimulus. And this final condition was basically so that we could examine whether there were predictive templates for expected stimuli that were not actually presented. So we performed very similar analyses to what was in the Peter Koch paper. So we trained pattern classifiers to discriminate between clockwise and counterclockwise gratings again, and we did this separately for expected and unexpected trials. And we find similarly that there was a superior decoding accuracy for discriminating between the orientation in expected trials than unexpected trials, which suggests there's slightly more information in the signal regarding these expected events. And then we also ran a kind of univariate stimulus-specific analysis in which we separated voxels that were tuned to the presented stimulus or tuned away from the presented stimulus. And this kind of analysis allowed us to look more closely at this sharpening effect to see if we could find any additional evidence for it. And so we find this kind of expectation suppression effect only in the voxels tuned away from the presented stimulus. So here, expected activity is lower than unexpected activity, but only in the voxels that weren't tuned towards the presented stimulus. So that this again provides similar evidence for the sharpening account. And then finally, we looked at the emission trials, and we looked at voxels that preferred the expected outcome versus an unexpected outcome, and we find that there's more activity relating to voxels that have the prefer the expected outcome. So when participants made an action, what they expected to see, but then didn't actually see because it was omitted. So this suggests that there is a predictive template for stimuli that are expected but not actually presented. So just to kind of conclude, these findings are then in line with the sharpening account that representations can be sharpened and even pre-activated when they're expected. And it's kind of maybe a little bit less consistent with cancellation accounts, because although we observed this expectation suppression, it was only in the units tuned away from the presented stimulus, so it could be which is more likely represented under this sharpening account. This finding is kind of consistent with some of the behavioural evidence from our lab that we've observed over the years. For example, visual perception is biased towards perceiving expected action outcomes. Auditory tone durations are biased towards the expected action durations. Expected visual action outcomes are perceived with higher contrast, and expected tactile events have been found to be perceived to be more forceful when they're predicted on the basis of action. And so these findings are then more consistent with this kind of Bayesian or upweighting accounts of perception. And we don't really find any evidence to suggest that action predictions do have a distinct cancelling influence on perception, as the evidence kind of is more consistent with that outside of action as well, so suggesting that predictions operate similarly across domains. Yeah, thanks for listening, and thanks to my collaborators and funders. I can't hear you.
Sonia, no tienes el audio.
Sorry. I was saying thank you to you for this presentation. It was really, really interesting. And now we have some minutes from questions from the audience. And we have here one question for you. They're asking, comparing expected versus unexpected as with self-generated versus externally generated stimuli may be different than comparing congruent versus incongruent stimuli? Or do you think that the same mechanism underlie both processes? For example, self versus externally generated and congruent versus incongruent?
So we've actually done a bit of work that we think that it's possible that when you make some like the self versus externally generated comparisons, it's possible that some kind of sensory gating mechanisms could be underlying these kind of effects, not necessarily explaining everything, all of the predictive accounts of them, but more that when you compare kind of action versus no action, the sensory gating is more likely to be present in the action condition versus the passive condition. So the reason why we compare expected and unexpected events is more so that we can always look at the patterns when they're always performing an action. So there's not that difference between the conditions. They're always performing actions, and then we also explicitly know that they've learned expectations. So they've learned to expect particular stimuli because we've trained them before. So we just kind of think that this expected versus unexpected is more like specifically looking at the prediction mechanisms rather than controlling for any other potential other influences that we can't control for in other comparisons.
Okay. We have also time for another question. They're asking you do you think that studies that have previously found cancellation effects could be wrong? Do you think that it's maybe a matter of how we analyze the data?
Yeah, I don't think cancellation effects are wrong. So I think like in terms of this study, so the point kind of is that some of these expectation suppression effects that we've observed, if we just look at univariate effects, it might not be telling us the full story. So an actual suppression in the activity might not be telling us the full story in terms of you can actually find more information about the expected signal within this suppression effect. So yeah, I think it's more of a matter of how we analyze the data in terms of these neural effects.
Okay. So I think we don't have more questions. No. So see you later. Emily,
Yeah.
Thank you so much for being here.
Thank you.
Bye. Now, help me in welcoming our last speaker from this first session, Dr. Anna Marcekova. I hope my pronunciation is correct. Dr. Marcekova is a postdoctoral researcher from the Tom Bergut's lab at the Ghent University in Belgium. She got her PhD in 2017 from Leipzig University in Germany, investigating how attention and perceptual expectations interact to shape visual perception. Today, she's going to talk. Hello, Anna. Thank you for coming today. I was saying that today you're going to talk about pupil-linked neuromodulation and temporal dynamics of expected and unexpected uncertainty. It means how we orientate in an uncertain world. So I hope you can share your screen.
Yes, I'm trying to share my screen now.
Yeah.
Hopefully it works. Yes. Can you see my slides?
Yeah, perfectly.
Okay.
So,
perfect.
Whenever you want.
Can you hear me well?
Yeah, we can hear you.
Okay, perfect. So thanks a lot for inviting me to this symposium, and thanks a lot for organizing this wonderful symposium. It's a real pleasure for me to celebrate this day with you today. And I would like to walk you through my presentation on pupil dilation correlates of expected and unexpected uncertainty, which is part of my postdoc project at Ghent University. And to introduce the topic for this, I would like to invite you to come with me to pre-corona times, when many of us used to travel a lot and move around a lot. This is a photo of Paris North Station, which is one of the busiest station in Europe. And the question I'm asking in my project is, how do we orient attention in such a complex and uncertain environment? Very often it happens when we travel by train that we arrive to the platform and our train is delayed and we are uncertain about our precise arrival time. We have to stay at the platform and we search for the displays for the announcement. It also happens that sometimes we arrive to this platform to realize that our train is cancelled and we have to leave the platform in search for other modes of transport. In this work, I'm proposing that attentional orienting can be defined as probabilistic inference about relevant spatial context based on such unreliable information and in ever-changing context. And in my work, I'm using probabilistic Bayesian framework, which can be used to estimate uncertainty of these inferences. An influential computational framework by Angela Yu and Robert Dayan proposes that our brain estimates two types of uncertainty. Expected uncertainty refers to the unreliability of the predictive relationship within a familiar context. So for instance, unreliability of our train connections on our daily commute to work. This type of uncertainty is thought to be signaled by cholinergic system. On the other hand, the unexpected uncertainty refers to gross changes in the environment, which leads us to assume that the context has changed in search for other alternatives. So for instance, train cancellation due to sudden snowstorms, which happened recently in Germany. This type of uncertainty, uncertainty related to these gross unexpected changes is supposed to be signaled by a noradrenergic system. In the current study and in the current project, I'm trying to dissociate behavioral consequences of expected and unexpected uncertainty, and I try to link it to the computational model estimates. Importantly, we are also trying to probe the links between neuromodulatory brain systems and these uncertainty estimates. For this, we are using pupil size, which is a proxy traditionally understood as a proxy for a noradrenergic signaling. In this work, I'm using two measures. It's pupil diameter and temporal derivative of the pupil. Pupil diameter has been shown in previous animal work to be correlated both with cholinergic and noradrenergic system, whereas the temporal derivative of the pupil was shown to be more strongly correlated with the noradrenergic system. To study these two questions, I'm presenting participants with an experimental task in which I present them with visual cues, which point to the left or right side of the visual screen. Participants are then presented with a stimulus, and their task is to respond to the spatial location of the stimulus. One of the cues is always relevantly predicting and correctly predicting the upcoming target location. So we call this relevant cue, and this cue is validly signaling the upcoming target in most of the trials. On the other hand, there is an irrelevant cue, which is predictive of the target non-predictive of target location. So it's predictive of the target location only at the chance level. This is an example of a valid trial where the relevant cue correctly predicts the target location. In the invalid trials, the relevant cue incorrectly predicts the target location. We manipulate unexpected uncertainty in this task by changing the identity of the relevant cue. So we call this cue switches. Cue switches are inserted in unpredictable moments in this task, and participants' task is to infer the identity of the relevant cue and then to follow the cue so that it helps to respond to the targets. Expected uncertainty, on the other hand, is manipulated and refers to the reliability of the cue, so how many of the valid and invalid trials are in the task. In the high expected uncertainty condition, it's a 70% cue validity, whereas in the low expected uncertainty condition, it's 85% cue validity. Importantly, we try to derive trial-by-trial model estimates of expected and unexpected uncertainty by using a computational model by Yu and Dayan. This computational model approximates belief distribution about currently relevant context and about the context change. So in this case, it's the belief about the currently relevant cue, which changes over the course of the task. The contextual change is assumed where the level of unexpected uncertainty crosses a threshold given dynamically by expected uncertainty. We can also derive prediction errors from these models, which combine the uncertainty estimates, so estimates of expected and unexpected uncertainty, so that we arrive at the trial-by-trial measure of prediction errors. I will now walk you through the data. So with the behavioral data, we have collected data from 29 participants. We have collected pupillometry data and also electroencephalography data. And I will talk today about the behavioral and the pupillometry results. So the computational model predicts that our attentional orienting will be less efficient under high levels of unexpected uncertainty. So we can see that the validity effect, which is often understood as the signature of attentional orienting, and refers to the contrast between invalid and validly cued trials, this validity effect gets much smaller under high unexpected uncertainty. These are the predictions of the computational model, so the dependent variable are prediction errors. We observe this pattern also in the behavioral data, so in the response times of participants. We observe this linear decrease of validity effect, so the linear decrease in the efficiency of attentional orienting with high level of unexpected uncertainty. The computational model will also expect these effects for this effect for expected uncertainty. So the linear decrease of validity effect. We haven't observed this interaction, but what we see is that specifically on valid trials, the response times slow down as a function of both uncertainties. In the pupillometry data, we actually also observe a strong validity effect. So the phasic target evoked pupil diameter is much larger on invalidly cued trials than on validly cued trials. More importantly, we are interested in how this pupil diameter varies as a trial-by-trial estimates of expected and unexpected uncertainty, which are derived from the computational model. And here what we can see are the beta coefficients from linear mixed effects models at each time point. And we see that indeed the phasic pupil diameter tracks these levels of expected and unexpected uncertainty. It decreases with expected uncertainty, and it increases with unexpected uncertainty. When we specifically look separately at invalid and valid trials to tease this out, we see that increasing pupil diameter with unexpected uncertainty is mostly observed on valid trials, whereas this decrease in pupil diameter with expected uncertainty is mostly observed on invalid trials. We also analyze the pupil temporal derivative of the pupil, also the phasic evoked response to the target. And here we also observe the validity effect. So the temporal derivative of the pupil is larger for invalid versus valid trials. And what we can also see, it varies as a function of unexpected uncertainty. So it's larger at a certain time point for higher values of unexpected uncertainty. On the other hand, we don't see a rather robust relationship with expected uncertainty in the pupil derivative. So I think we can conclude from these results that attentional orienting relies on Bayesian uncertainty estimates, because the model-based estimates of expected and unexpected uncertainty are reflected in the single trial behavioral responses. We observe slowing down under higher uncertainty levels, and the attentional orienting efficiency, which is measured by the validity effect, decreases linearly with increasing unexpected uncertainty. One could also ask about other modeling approaches, and we have approached this by fitting also other computational models, so from a group of reinforcement learning models, but we didn't observe significant relationship between response times and predictions error derived from simple Rescola-Wagner model or Rescola-Wagner with adaptive learning rates. Also, we have recently tried to put this and replicate these findings in another task. So we used a visual search task, which is much more complex and where we manipulated feature-based attention. With regards to the pupil size correlates, we can see that pupil size indeed tracks model-based uncertainty estimates. Phasic pupil diameter is smaller with increasing expected uncertainty. This is mostly on invalid trials where we predict that this might be scaling with surprise, as the invalid trials are actually less surprising under high expected uncertainty. And the pupil diameter increases with increasing unexpected uncertainty. So it seems that the temporal derivative of the pupil tracks these unexpected uncertainty estimates. And I think this dissociation may also point indeed to the involvement of different neuromodulatory systems in the uncertainty estimates. So we have observed that these two types of uncertainty are differentially reflected in pupil size measures. They have also dissociable time course. Expected uncertainty effects we observed a bit earlier in the TARCOS, and these effects were more sustained, whereas the unexpected uncertainty effects were later and they were more phasic. We didn't observe any significant effects in the baseline pupil size or robust effects in the Q target interval. We are now currently linking this pupil diameter indices of uncertainty with EEG signatures of attention, and we will try to further dissociate these two distinct types of uncertainty in attentional orienting. With this, I would like to thank you for your attention, and most importantly, I would like to thank my co-authors, Tom Verhoets and Eva van den Busse, and Ghent University and Vrije Universiteit Brussels and Research Foundation Flanders for the funding for this project. I'm happy to take any questions.
Thank you, Anna. I think that Sonia's connection now is the one that's not very good. We would like to ask the audience if they have any questions to put them up on the questions and answers pop-up, or directly ask them on the chat, because right now, I think it was a great study, but it was a little bit complex, and I think that people are thinking about it right now.
Can you see me now? Sorry.
Yes, Sonia, it's perfect now.
Thank you. Like my connection now didn't want to work. Thank you, Anna. I was really interested. Can you see and hear me?
Yes, I can see and hear you. Yes.
Okay, it's like my screen is totally blocked. Like I cannot see anything. I see that there are. Okay.
Can you see it, Sonia, or I read it? Okay, there's a question for Anna. And they are asking, like, what would you expect to observe in your EEG data?
Yes, indeed, that's a very interesting question. We actually started to look at it, but I thought there might not be enough time to talk about all these issues. So what we are looking at in the EEG is, first of all, the P3 component, which is very often linked to the pupil dilation. So we try to tease out that, and we have already observed the linear relationship between the P3 component and the pupil dilation measures. And another part which I'm looking at are the oscillatory signatures. So we try to look at the alpha oscillations, which we thought, based on previous research, may be linked more to expected uncertainty, whereas the theta oscillations, which we expect to be most related to the unexpected uncertainty.
Thank you. Sonia, can you see now the question panel, or not yet? Not yet. Okay. Okay. Right now there are no more questions for you. I'm going to give the word to Sonia so she can close the session. And I think that if anyone has more questions, they can put them on the chat, so you can go there and answer them later. Maybe when people have time to process, they will have more. Thank you.
Thank you so much, Anna. So sorry, now if you're writing, I cannot see it, but I just want to thank you all for being here in the first and the earlier session of our virtual symposium, and I hope that you all have enjoyed and learned some interesting things in this first session. We now have a short break until 11 o'clock when session 2 will take place. Please remember to close this session and log into the session 2. You can go directly to the second session by clicking schedule on the top left part of the website. So goodbye for now, and see you there in some minutes. Okay? Goodbye.